All right, everyone. Thank you very much for being here with us. I would like to introduce FeMS very quickly in 1 or 2 sentences in case some of you don't really know who we are. Females in mass spectrometry. I mean, basically, we are welcoming anyone, men and women, even though we are called females in mass spectrometry. I should start with this. I think that very much everyone is welcome. And I could try to share my screen with you and show you the website of FeMS. Just 1 minute so that you can see. So we have here our website. And basically, we are a community of women internationally located and volunteers to support women in the field of mass spectrometry. And we do a lot of different things. We have events like you are joining today. We do them online, but also in person at conferences. We have many awards for a different level of people, as in early career or later career. And I really invite you to check them out because some are really nice and I would say unique in that sense. We have resources with directories of underrepresented scientists. We do mentorship and yeah, we have a lot of tips and you can also post jobs and we have a job board. So if you're looking for a job or you want to recruit someone, go ahead, etc. I will not spend more time on this. But I just want to thank our sponsors as well, because without them, we would not be able to do everything we're doing. So I will stop sharing my screen if I manage to do this. Yes, great, we're back. And I will introduce our first speaker, Behnaz Akbari. She used to work at Michigan State University, and I have understood now that she's going to work with the Martin Luther Center in Boston University, at least for some time. And she will talk about recent advances in neurodegenerative and neuroinflammatory diseases using mass spectrometry. Go ahead, Behnaz, you can share your screen.
I'm trying to change the, yeah, it's good now?
Yes, you're fine. Go ahead. We can see it.
Okay. Hello, everyone. My name is Behnaz, and today I'm going to talk about recent advances in neurodegenerative and neuroinflammatory diseases using mass cytometry. Before I start my talk, I would like to appreciate Julie and her team to facilitate everything and regarding the talks. And I know how difficult it is to handle all these things. So I just want to appreciate their team before I start. And here you see my education, my background, and what I would like to achieve is I'm a very, I should say, enthusiastic researcher. So I would like to expand my knowledge in the field of bioimaging and especially mass spectrometry and quantitative imaging techniques. And I would like to say my long-term mission is to establish a real-time human surgical decision-making. And based on my background, I would like to be also a multidisciplinary scientist. So I would be very happy to, I'm collaborating with several companies, but I would be happy to have more collaborations and do networking. So I think the topic has different pieces. And one piece is about the mass cytometry, that we should understand what this technique is. So the principle of mass cytometry actually is that it's all about a modified ICP tough mass. For those who doesn't know about mass spectrometry, it's a technique, it's a chemical tool, which is inductively coupled plasma time-of-flight mass spectrometry. It's a sort of, it's a type of mass spectrometry. It's a new, and mass cytometry is a new platform of this mass spectrometry. I mean, it's able to do subcellular resolution, and effective cell segmentation could be obtained. And there would be no autofluorescence when we do staining and we do some using, for example, some imaging techniques to track the molecules. And it is all about the flow cytometry, that it is connected to the mass spectrometry. So how it works, it's all about the the antibodies that are labeled with heavy metal ions tags, rather than using fluorophore that is used in flow cytometry. And it is read out by time-of-flight mass spectrometry. Because of those metals, we have a specific mass to charge ratio, so it's very easy to track them. And it's also the great part of this technique is that you can measure 45 so far 45 simultaneous. I mean, you can measure 45 simultaneous parameters without using fluorescent agents. It's very similar to MALDI MSI. Different techniques, but the basis is somehow is the same. And this allows for combination of more antibody specificities in a single sample without significant spillover between channels, as you can see here. And then then what we can do after measuring after the quantification of 45 metals that they are isotope, they are non-radioactive. Then we are using some computational tools. That this is the main part of this mass cytometry, because we have to just transfer our data, which is coming from the tumor tissue, blood or lymph node. Then we have to convert them to the databases. Usually they use cytobank. And with that, you will able to do lots of clinical biomarkers and discover lots of discover several new biomarkers, and realize that what molecular pathway, for example, is in a disease exists. So this is the history of mass cytometry, and as you can see, this has been started from 2001, and it is still ongoing. And then, as you can see, it was first started by Yang group, and then mostly the books and publication says that it's about the Tanner group, that they have been able to use the stable isotope nanoparticles in order to use mass cytometry for immunoassay. And then it has we have reached to this type of mass cytometry. Again, it's coming from Tanner's group, that they are, I think they are in University of Toronto. And then the most popular protocols are coming from the Stanford University and Dr. Nolan, Gary Nolan. So they have so many fascinating protocols, and they are working on human patients. So the principle of imaging mass cytometry, again, this is the same technique, but it is all about the imaging. And what it is added is about the laser, that we use a laser in order, especially with a pulse 230 nanometer, and it will raster scanning on the tissue, as you can see here. And then again, the same procedure, meaning that the metals will add it to the antibodies, and then you will see the abundance in this technique, imaging mass cytometry. And then, as you can see here, this technique, what is the difference between CyTOF and imaging mass cytometry is that you can find in gray scale, as you can see here, you can find anatomical details, and which is very interesting, although it is ex vivo, or sometimes it's just, for example, we can use mouse brain to find such a amazing technique, amazing images, but actually this is how this works. And it is very important to understand that the spot size is only 1 micrometer. So that's very, very important, because it's just it shows that it's just a very, very small part of the region of interest can be measured. So another technique that has been introduced by Nolan and the research group is multiplex ion beam imaging mass cytometry. And it is all about the secondary ion mass spectrometry that's used. And as you can see, it is a raster on the the slide, and then it can measure the antibodies that have been tagged by metal isotopes. So, and then you can see also the real instrument here, that how this works. And actually the important features of this is that visualization of 40 markers simultaneously, imaging at the subcellular, that other mass cytometry, imaging mass cytometry and CyTOF cannot do that, and detect of low abundance protein, and re-scanning of the slides. So that's important. So with that in mind, due to the time, I want to be very considerate about the time. So so CyTOF experimental procedure, you can see I'm giving you the whole picture of the experimental procedure of CyTOF. I'm only telling you the main part of this technique. We have to consider the method, what type of method, and what patients we are going to work. And then we have to do acquiring data by barcoding, meaning this is a metal tag and reference sample. We have to have reference sample, staining with a standard antibody cocktail. Cocktail means just the antibody plus metal, and instrument calibration. And then we have to do some do a computational as you can see here, we have to do some several computational analysis that can answer 1, 2 or more biological question. That's so important, that what questions we have. So and then at the end we analyze the data, and then we get the results and the work on for example biomarker discovery, or prognosis prediction, and so on. So now let's go to the application of CyTOF in human diseases. So mass cytometry can be used in cancer, autoimmune disease, immunophenotype, and also other, like pharmaceutical science, or drug delivery. So it can be very useful for many diseases. So, yeah. So, but here we want to focus, and mostly the publications are all only working on breast cancer, but here we only want to work on brain, and how we can get so many salient features of brain. So here you can see that the type of central nervous system that is important to understand. We are studying neurodegenerative diseases, glioma, and how the axons or neurons are damaged. So this is the whole story of neurodegenerative and neuroinflammatory diseases in one picture. And here you can see the real, actual laws of neurological function. As you can see here, that there is a plaque of demyelination in this part of, it's very close to the white matter, and it says, it shows how it is has been just damaged. And we are going to work on this part. So CyTOF is able to give us a small number of immune cells that exist in the brain. And under neuroinflammation condition, we are able to understand the phenotype, functional, and activation markers of various immune cells, as you can see here. So I chose only two studies. I will have to escape some of the pictures because each paper had around 60 pages data analysis. So I have only chose some of the important data, and one of them is single cell peripheral immunoprofiling of Alzheimer and Parkinson disease, that they use this CyTOF, and they use this this because there is no blood based clinical biomarker for AD and Alzheimer and Parkinson disease. They have used this CyTOF, and they were able to use the blood samples in order to find the neurological dysfunction. And the problem was that previously they have recognized that there are so many variation, and when they are using this blood samples for especially for a specific site of these neurons, and that's why it would be very challenging. So that's why they decided to use the blood samples, but using this use the cohort studies, and it's a very powerful stimulant of innate immune response that activates via a toll-like receptor. So these are all the important biomarkers of Alzheimer and Parkinson diseases that they have worked on. So they have been labeled, and then they were able to use the computational analysis in order to see how what type of cells, neuron cells, are correlated to each other, and if there is to determine, for example, which components of peripheral immune response correlated with diagnosis of AD or PD. And again, this is another data analysis that we can find the correlation network based on the FeMS technology, computational platform, and we can understand the immune features obtained from CyTOF data, and to understand if it is healthy patients, if it is related to Alzheimer, or if it is related to Parkinson diseases. And we have another study, but I think we don't have so much time to consider, so I have to skip that. And so, summary. It's all about the neuroinflammatory and neurodegenerative diseases, which is included Parkinson, multiple sclerosis, Alzheimer, Huntington diseases, and the migration of peripheral immune cells in this, especially when we want to work on pathological aspects of any disease. So CyTOF is very powerful, expensive technique, but the good point is that there is no special overlap, and it's very detailed and precise, and it's possible to use it in brain diseases. So the outlook would be development of vaccine or drug, advance the research in the clinical immunology, and uncover many questions about immunopathogenesis and immune response. And I was thinking about multimodal imaging protocols to just the breadth of imaging mass spectrometry with much more sensitive aspects to be considered. And it is also great for lipid analysis in the brain tissue for diagnosis of different diseases. And also maybe it's good to introduce, it's a time for the real test biomarker actually. So I want to dedicate this talk to the brave women in Iran that are fighting for their rights and freedom and given their lives in order to bring peace, justice, and prosperity to the country. These women are fighting empty-handed against the brutal regime that has been brutally killed and arrested a number of people as persecuting protesters. Please be their voice. Thanks for your attention.
Thank you, Behnaz. And yes, we are standing together with women in Iran. It's, I mean, this is not the topic of today, but I totally understand your feeling. I would like to, I mean, I could ask questions, but obviously if someone in the crowd wants to ask a question, just please. Bini, do you want to?
Hi Behnaz. That's a very
Hi.
nice talk, and I'm really curious about CyTOF of late, and I was researching on it, and it's good to have have some heads up on that. So my question is basically, there is a lot of single cell imaging mass spectrometry going on. So what is the decision point where you decide that, okay, I should do a single cell imaging mass spectrometry versus a mass cytometry? What makes you to take a call for one or the other?
Thank you so much for your question. It's very interesting. Imaging mass spectrometry can measure the region of interest between 0.5 to 5 micrometer. If we can consider the the latest version of imaging mass spectrometry, but imaging mass cytometry is able to do subcellular analysis. And it's detailed. Yes, it is a very expensive technique, but it provides you very great insight to understand the subcellular surface, and it is less than 0.5 micrometer, and it is precise. So that's why I call for cytometry. But each technique has its own advantages and disadvantages. It depends on our biological questions, so.
Okay.
If I may have a very quick follow-up. I would assume that if the question is to decide between mass cytometry and the conventional imaging, super resolution imaging, I would consider that your argument is the background fluorescence, which is favoring mass cytometry over going with the fluorescent imaging. Am I right there?
Yes, you are very correct. And the point is that for mass cytometry technique, there would be no challenge regarding autofluorescence. But in other techniques, you have to consider, even there is a software that I'm going to work with Martino Center, but it's called Halo, H-A-L-O. That is wonderful. It helps to just consider the autofluorescence. So autofluorescent molecules, however, it's not that much precise. It's just good for patients and surgeons to make quick decisions regarding, disease, especially brain disease or any ex vivo tissues. But imagine how this is, how this could be, helpful in this context.
Thank you very much.
I'm looking at the time, and of course, I was responsible for some of the delay, but I think we should move on. Thank you very much, Behnaz. And I will introduce our next speaker, Neena Susan Eappen. Yes, that's you. You're a PhD student. You're working at the University of Toronto, and you will talk about developing mass spec techniques to study intrinsic structure of peptides and proteins. And I think that you have an interest in moving to MS industries focusing on clinical diagnosis, if I'm not wrong, but I'm pretty sure you will tell us about this. Go ahead.
Is that good? Can you see my slides?
You're good. Go ahead.
Okay, thank you. Thank you so much to the entire FeMS team for this amazing opportunity. So I'm going to present a bit of my thesis summary here, what I've been working for the past 6 years. So I'm basically developing mass spec techniques to study intrinsic structure of peptides and proteins. So a bit of what I've done so far, I did my bachelor's at UFT, double major in biochemistry. I'm currently pursuing my PhD in analytical chemistry under the supervision of Rebecca Jacques. I think some of you know her in this audience. Sorry, let me just switch to that. Yeah, I'm expected to finish in March 2023, so I'm really close to finishing this soon. So my research in Jacques lab is looking at intrinsic structure. So why I'm interested in this? Because so we know in solution native structure is stabilized by both intra and intermolecular interaction solvent. So I'm interested in looking at what happens when you remove the solvent. So when we transfer to gas phase, there is no more hydrophobic effect. There is a lot of like enhanced electrostatics. So the question is to what extent native structures retain in gas phase. And in Jacques lab, we look at gas phase conformation using different techniques. My focus is on a technique called gas phase hydrogen deuterium exchange. This is a typical technique in solution phase. So where hydrogens bound to heteroatoms like nitrogen and oxygen, they are getting exchanged by deuterium via proton transfer reaction, the deuterated reagent. Because I want to probe conformation of gas phase structure, I do this reaction in the gas phase using like deuterated water or deuterated ammonia. Why am I particularly looking at this technique? This technique is not well understood. The rules regarding this technique, so like where and how fast exchange happens depends on whether those labile hydrogens are protected by hydrogen bonding, let's say in secondary structure like alpha helices, how accessible the reagent is to those sites, and like so many things like gas phase basicity, location of charge sites. So there are just so many factors affecting this gas phase HDX. So in my thesis, I have 3 main chapters where I investigate this technique in different systems. So I'm going to give a quick summary of all 3. So my project 1 is trying to understand the factors affecting gas phase HDX. So I'm looking at this series of peptides, alanine-based peptides. It's a polyalanine, so there are 15 repeats of alanine, as you can see in the sequence. with lysine and arginine in different positions. I have like position 1, 7, 10, and 16. So here I've shown a sequence of, let's say, the lysine at position 1. So it's called the K1 peptide. I first characterized them using ion mobility and some computations before HDX. So my IMS data showed that the collision cross-section was very similar for the arginine and lysine analogs. So at the same position, they had very similar collision cross-sections. And my computations showed that as the lysine or the arginine moved from N to C terminus, it transitioned from like a charge-solvated globular structure to a helical structure. All the inspiration is from Martin Jarl's work, but I was also just happy to see my computations also supported it the same. So when we applied it to, when we applied gas-phase HTS to these systems, I'm starting with the lysine analogs. Shown here are structures I computed using molecular dynamics. So we see here that, again, as the lysine moved from N to C terminus, the peptide became more helical. On top of that, there is just less HDX as it went from N to C terminus. We were able to assign where these exchanges were. So for example, for this particular variant, the K16 variant, those 3 exchanges, given all those amides are protected in the alpha helix, we assigned those exchanges to happen on the lysine, the pink residue. And for the other 3 variants, more amides opened up, there is more exchange there. And in terms of where the other exchanges are happening and the progressive trend, we found that the proximity of this lysine to N terminus was key. That's why there was more exchange as it went from C to N terminus. And we were able to relate this back to previous examples in literature, talking about some relay mechanism with ND3, where when we have 2 sites of similar basicity, there is a higher chance of better complexation with the ND3 molecule and thereby more exchange. So clearly, location of charge played a role in mediating exchange here. What is interesting in the arginine analogs is that there is like absolutely no exchange, like 0 deuterium uptake from the arginine analogs. And here again, my computations really helped understand this data. So here I'm showing the lysine and arginine analogs at different positions. On average, it is shown that these arginine analogs were less helical, and it is easier to explain given that arginine has a lot of like charge solvating opportunities. It is rendering the peptide less helical, but we still saw like 0 exchange from these arginine variants. Even more striking was the position 16. They looked exactly similar, but there is no exchange from the arginine variant. And we were able to explain this using, again, previous arginine literature, that it has, now that arginine is like highly charge solvating, it has enhanced gas-phase basicity, and there is like mechanistic implication that there is like lack of salvage intermediates, not allowing exchange. So clearly, inclusion of arginine did inhibit gas-phase exchange in these peptides. What I'm really proud of this project is that I got these computations running. And I was also able to apply this to my future projects. My project 2, again, I looked at the same technique. Here, my goal was to see how well can gas-phase HDX separate conformational isomers. So here, my system is the same lysine variants, doubly protonated now. So 1 charge is on lysine, and the 2nd charge assigned to the N terminus. So I'm just looking at 2 positions, position 1 and 16. I've characterized it first using ion mobility. So using ion mobility, we saw like 2 drift time populations. Yes, one a compact species and an extended species for both the peptides with like contrasting relative intensities. Even more interesting for the K1 peptide, when I attended IMS on different days, multiple trials on the same day, the distribution varied for K1, even with the same instrument settings. And we tried some source activation experiments. Even further down, we were able to rule this or assign this to some like humidity or temperature changes in the source region. And this is usually this usually indicates existence of peptide protomers. So we believe there is some kind of proton transfer that happened during ESI or upon ESI. And I was able to confirm this using computations, existence of protomers. So I got some computations running where for my K1 peptide, I kept one of my protons still at N-terminus and another version with the proton at the C-terminus and an amide oxygen. And I was able to support the IMS data where I have protomers in K1 and I just have different conformers of the same protomer in K16 based on relative, the differential collision cross sections and relative energies. I was able to assign these populations to the IMS data. What happened when I had tinted gas-phase HDX on these peptides? So here I'm showing like the raw HDX data, like the mass spec data extracted for the K1 and K16 peptide. I was clearly able to see like two populations. So I'm showing like the average digium of peak there. They differed by like 5 hydrogens. So clearly like two predominant populations were seen in both K1 and K16 to a less extent, but like a big summary of this project is that gas-phase HDX is able to separate conformational isomers with two distinct exchange rates, a slow and fast exchange population. It is consistent with the IMS data. What I'm really proud of this project is, again, fine-tuning HDX to see these multiple populations. I had to figure out multiple conditions to get these two conformations. And again, the overall computation, really proud of these computations running. And my third project, I'm looking at the same technique along with another sequencing technique called electron capture dissociation. And here I'm characterizing a protein, slightly bigger, but it's actually one of the smallest proteins. It's called tryptophan cage. This is the structure. It's stabilized by like a hydrophobic cage and salt bridge, and it has an N-terminal alpha helix. So I'm looking at the wild type and a series of mutants. The wild type is not very stable at room temperature. Mutants at position 9 and 10. Position 9 is known to be structurally more compact in solution. At position 10, it's more thermally stable. It's actually the D-alanine version. And I have a control variant, which is again at position 10, the normal L-alanine mutation. And it is in solution, it is a random coil, totally unstructured. And I confirmed with my own circular dichroism results. And I also did some thermal stability experiments. So the extent of helical character was found to be the least in the control, as expected, and then the wild type, and the most in the position number 10. Okay, and this protein exists as a zwitterionic 1+ in solution phase. So in gas phase, when I attempted gas phase HDX, I did on both 1+ and 2+. 2+ because it was the most predominant charge state, and on the mass spec. So one of the surprising results was that both the 1+ and 2+ gave just 40 to 50% of exchange. So it is pretty much protected in gas phase. So we were happy to see that, okay, all the 4, including the control, the random coil in solution phase, appear to be structured in gas phase. So we believe that there is some kind of refolding that happened to this particular variant upon desolvation, which we have seen with several other proteins in our lab, where the protein refolds in gas phase. In terms of extent of protection, more or less the prime did match up with the solution phase thermal stability results. So there is some structural memory upon desolvation, except for the D9E variant, or the 10 and 9 positions were switched the order, we believe, because like gas phase HDX is more sensitive to the global protein structures and more to the structural compactness. So we know from literature that position 9 is more structurally compact than the G10DA variant, but circular dichroism is not sensitive to it. So this is one thing we were also happy to see, that gas phase HDX is sensitive to these, the big picture, not seen in solution phase. And when I attempted electron capture dissociation of the 2+ variant, of course, I cannot do it of the 1+. Here I'm showing like the extent of fragmentation sequence coverage from the protein for the wild type and the 3 variants. There is like poor sequence coverage from the center of the protein, which indicated that there are like strong intramolecular interactions in the protein, and the protein is structured in the gas phase, again supporting and complementing the HTX data. And the other thing, the control variant had the exact or very similar fragmentation profile as the wild type and the other structured variants, again supporting that it did refold in gas phase, indeed, upon desolvation. And the extent of fragmentation was very much in trend with the solution phase thermal stability results. And we believe that these trends are more consistent because my sequence coverage is very, it's actually probing only the N-terminal helical region, which is exactly what circular dichrosis is probing, the helical content. So that's why I think the results were consistent. Regardless, a big summary of this project was that gas-phase HTX and ECD are complementary and they're sensitive to all these structural differences. I'm really proud of working out reproducible HTX and ECD experimental for this project. It took really long. So a big picture summary of all my projects is that I investigated gas-phase HTX in multiple systems, and I checked how complementary it was with like techniques like ion mobility, electron capture dissociation, and supported them with computations. What do I want to do next? Definitely not academia. I want to transition to industry. My research interested proteomics. Yes, I do not have any proteomics experience, but I've definitely dealt with complex data such as ECD, adding on that with HTX, so much of signal dilution. So I've definitely dealt with complex data. And I really want to try out new, take up new challenges like proteomics. I'm really interested in MS instrumentation, protein structure characterization, as you just saw, and biomarker identification. In terms of my skills and expertise, worked tons with nano-ESI. All my experiments were nano-ESI and on a high resolution FD-ICR. All my calculations were done using GROMACS package. I'm experienced with ion mobility. Not a lot of solution phase HTS, more on the gas phase HTS side, electron capture dissociation and circular dichroism. What I really enjoyed during grad school is the instrument troubleshooting part more than my thesis. Yes, a lot of downtime during instrument troubleshooting, but just isolating that software versus hardware, even with that hardware, is it power supply or is that little component here and there? That is like the best part of grad school that I enjoyed the most. For all my projects, so our lab is like mostly a spectroscopy heavy lab. So in my projects, I didn't have any spectroscopy, so I had to reach out to different collaborators, different labs to get insights from like various parts of my project. In terms of my timeline and where do I want to work, I really want to settle in Ontario, and I'm available to work anytime from January once I'm done submitting my thesis. And thank you again, FeMS, for this amazing opportunity and everyone for listening.
Thank you very much, Neena. Do we have a question from the audience?
Hi,
Bini again, go ahead.
Yes. Hi, Neena. Nice talk. I might have missed this point. What's the comparison between liquid phase and gas phase HTX? Is there something which is specifically advantageous to go ahead with the gas phase HTX?
Yes. So
and a quick follow-up, I can. A second part of the question is, how is it comparing with the other, the HDX kind of techniques like HRF and that photo FPOPs and all? How is it the gas phase HDX in comparison to those methods?
I'll take the first question first. So in solution phase, so you have different types of labile hydrogens. One, really fast exchanging on the N terminus on the side chains versus slow exchanging on the amide backbones. So in solution phase, you cannot trap those fast exchanging ones. They're too reactive, whereas in gas phase you can. So I'm able to probe all those fast and slow exchanging hydrogens. For your second question, I actually don't know. That's a very good question. I should prepare for my defense. How does it compare to FOP? I know FOP is an oxidative link reaction. Maybe it's faster. So the lowest time scale I can do these reaction times I can expose my peptides to the deuterated reagent in gas phase is 5 milliseconds. I'm not sure if FOP, maybe FOP could go faster than that. I'm not sure. And I believe FOP happens earlier on in terms of labeling process, to my understanding. But for my experiments, I do accumulate my peptides and proteins for like hundreds of milliseconds in gas phase before exposing them to the reagent. So maybe there are more like structural rearrangements happening before my gas phase exchange experiments. Maybe FOP is more can probe the native structure better. That's a wonderful question. I should prepare that for my defense. Yeah.
I have a
Thank you.
very quick question, but we need to, and then we need to move on. Because you do a lot of computational, it looked like you really enjoyed doing the computational part of things. But what type of language did you use? You talked about GROMACS package. Is that, what language do you use to compute your calculations?
I'm not exactly sure how to answer that.
I mean, would you be basically? Would you be?
It's a Linux. It was a Linux interface, so.
Okay, no, I just pictured it because I'm a metabolomics person, so I was thinking Python, R, stuff like that, and I don't know if I just thought that if it's something, because it was not explicit in your presentation, but if it's something that you like and that you know how to do, I just thought.
I'm not. Yeah, I'm not used to like all those R, Python. I did not have any of those skill sets, but in GROMACS we had like this PuTTY or Linux interface. Yeah,
Okay.
this is regular Linux commands, which were which were not straightforward for me to begin with, but for me now, I'm very comfortable with that.
Good. Okay, thank you, Neena. It was not a trap question. Okay, I will move to our final speaker, Lisa David. And Lisa, you're a postdoc. You work at the North Carolina State University, and you will talk about discovering mobile signals involved in plant immunity.
Yeah, I can start to share my screen now.
Yes, please go ahead.
Let me just put this into presentation mode.
Great.
And I want to put my pointer on for you. Yeah. All right. So yeah, I'm Lisa David, but thank you all for being here and giving me your attention today. I'm just going to tell you a very short story about mobile signaling mechanisms involved in plant immune responses. And I know that a lot of people here are not plant biologists, so I'll include some background examples for you. But I just want to use some examples from my PhD work and also a little bit from my postdoc, but mostly from my PhD background. So I'm just going to start out with giving everyone some background about myself, because I know not everyone has been able to look at my LinkedIn page yet. So just briefly introducing myself and telling you about my background. And then I'm going to give you a very general overview of plant immunity, and I'm going to focus on 2 specific plant immune responses. The first is this systemic acquired resistance, or SAR, which is what I studied during my PhD at the University of Florida in the lab of Dr. Sishu Chen. And then I'm very briefly going to talk about the research I did as a postdoc at North Carolina State University, which involved induced systemic response, or ISR. And I'm going to conclude by discussing some of my career plans for the future. So yes, I received my PhD from the University of Florida last year, and my dissertation was entitled The Effects of Systemic Acquired Resistance on Guard Cell Immunity. And I have 5 first author publications, 3 co-author publications, and 1 other first author publication that's in process from my PhD research. After graduating, I started my postdoc at NCSU in July of 2021, and just now I've accepted a new industry position in Research Triangle Park. I also have more than 10 years of experience as an educator. After my master's degree, I taught high school and community college courses, and I was also a teaching assistant throughout my entire PhD, where I mostly taught the laboratory courses for the biology department. Also, during my PhD, I mentored 10 undergraduate students, and this has been one area of my career that I've really found to be the most rewarding for me. I have also been involved in a number of synergistic activities, but here I'm just going to highlight 2. During my PhD, I was a founding board member of the UF chapter of SACNAS, that's the Society Advancing Chicanos, Hispanics, and Native Americans in Science, and I held various positions during my time at UF SACNAS. I'm also currently a member, of course, of FeMS, Females in Mass Spectrometry, and right now I'm able to be a hub coordinator this semester. So today I'm going to talk to you about 2 different types of plant immune responses, and I'm going to start with a very general overview of both. So for my PhD research, I studied systemic acquired resistance, or SAR, which is shown here on the right. So SAR was originally documented almost 90 years ago now, and that term SAR, systemic acquired resistance, was first used by Ross in 1961. But SAR is an induced resistance response to the perception of pathogens by cells in the infected leaves. And SAR is dependent on increases of the biosynthesis of a plant hormone called salicylic acid, or SA. And increases of SA activates this cellular defense response that's controlled by a master protein called NPR1, or non-expressor of pathogenesis related genes 1. NPR1 is a master regulator of SAR response, and its activation triggers a cascade of downstream signaling events that include reactive oxygen species and MAP kinases, calcium signaling, and expression of these pathogenesis related, or PR genes. The left panel over here shows induced systemic resistance, or ISR, which is not mediated by SA. It's mediated by different plant hormones, jasmonic acid and ethylene. ISR is activated in root cells in response to their association with the microbes in the rhizosphere. That's an area that surrounds the roots. So the important message here is that both ISR and SAR result in a whole plant immune response, which increases the stress tolerance to herbivory stress, wounding stress, abiotic stresses, and a broad range of different pathogens. So mostly I'm just going to talk to you about SAR response, which is characterized by this translocation of long distance signals from the induced leaves to the distant tissues, the distant leaves, resulting in what's called priming. It primes the uninfected leaves. So some of these mobile signals have been characterized. They include methyl salicylate, a G3 phosphate dependent factor, azelaic acid, and dihydroabetanol. I studied during my research DIR1, but I don't have time to talk to you in depth about this today, but there's also this lipid transfer protein called DIR1, or defective and induced resistance 1, which associates with AZI1, this azelaic acid-induced one, and they act as a chaperone for mobile signals in the vascular system of the plant. SAR research has been conducted using leaf tissues, but it had not been investigated in guard cells And guard cells have their own specific immune response because the stomata in the leaves are like the front door of the leaves, and they are the first line of defense against pathogens. So during my PhD, I asked how these SAR immune signals were affecting the guard cells in these primed remote leaves. So I put in this slide for those of you who are not familiar with plant research. I'll just briefly review the specialized role of guard cells. So leaf surfaces are covered in microscopic pores called stomata. And many of you are familiar with the two main functions of stomata. So first, they allow for gas exchange, acting as an entryway for carbon dioxide and releasing oxygen into the atmosphere. The other main function is regulating the water movement through transpiration. And there's a pair of guard cells which control the stomata aperture. So for example, water loss through the stoma during drought triggers chemical reactions that signal water and ions to leave the guard cells. And as the solutes exit the guard cells, those pair of guard cells deflate. You can think of like two flattening balloons that subsequently then close the stomata. So guard cells are responsive to numerous environmental stimuli, and some of these may seem obvious to you, but I think some others may surprise you. So guard cells close the stomata in darkness and reopen the stomata in light, particularly red and blue light. They also close during drought to prevent the water loss due to transpiration, but they open stomata in high humidity. They respond to other environmental cues such as carbon dioxide concentration and temperature. But then in 2006, Shengyang He's lab at Michigan State University discovered that guard cells have an innate immune response to bacterial pathogens. In particular, they found that stomata close when the guard cells perceive a MAMP. It's a molecular signal called FLAG22, which is a 22 amino acid peptide from the flagella of bacteria. And the bacterial pathogen, Pseudomonas syringae pathovar tomato, or PST, was able to reopen the stomata due to a microbial toxin called carnitine. So pathogen entry into the host tissue is a critical first step to cause infection. These foliar bacterial pathogens like PST, the stomata are important entry sites into the apoplastic spaces between the cells, which those bacteria colonize. Historically, stomata were always considered to be these passive portals for entry of these pathogenic bacteria. But Shengyang He's group found that they're not passive. So PST infects tomato, that's what it's named after, but also many other horticulture and agricultural plants that are economically important. And also this model dicot species, Arabidopsis thaliana. And this pathogen causes the disease bacterial speck, and under certain conditions, it can cause ice nucleation in the leaves and kill the plant. So this is guard cell basal immunity. I'm going to start over here. It's triggered when this membrane-bound pattern recognition receptor, FLS2, perceives that FLAG22 conserved peptide, that 22 amino acid conserved peptide present in the bacterial flagella. And upon perception, FLS2 then associates with this receptor-like kinase and a receptor-like cytoplasmic kinase, BAC1 and BIC1, and they activate these respiratory burst oxidase homologs, ARBO, D, and F, and that's causes the production of reactive oxygen species, which initiates the closure of the stomata and prevents the bacteria from entering through the stomatopore and colonizing the apoplastic space of the leaf. But as a counter defense, the bacterial pathogen, Pseudomonas syringae pathovar tomato, PST, produces this toxin, carnitine, which is a biochemical mimic of the active form of jasmonic acid. That's jasmonic acid isoleucine, the plant hormone. and it competes for binding to the jasmonic acid isoleucine receptor COI1, and that promotes stomatal opening, and it also blocks stomatal closure by blocking the biosynthesis of salicylic acid. So since the discovery of this basal immune response, it has been well studied, as you can see here, but it's almost exclusively been studied as a direct interaction between the pathogen and the guard cell. So I investigated the biological significance of guard cell priming in Arabidopsis by infiltrating one mature rosette leaf with either with the PST or a mock solution. And after 72 hours, that opposite rosette leaf was then exposed to the PST. And as previously reported here, the basal immune response of the mock treated stomata, they close after 1 hour of exposure to that FLAG22 peptide from the PST, and then they reopened after 3 hours as a response to the carnitine. But surprisingly, the prime leaves did not exhibit that same stomatal immune response. They maintained this small stomatal aperture during the entire period of the PST exposure. And here you can see there's no significant difference in the stomatal aperture from the prime leaves taken at 0, 1, and 3 hours after the PST exposure. So, and then I measured stomatal immune responses. I found after 3 hours of PST exposure, significantly more bacteria entered into the mock leaves when I compared them to the prime leaves. And then I examined the overall susceptibility to the PST. I measured bacterial growth in the leaves after 7 days, and significantly more bacteria could colonize the mock leaves that when I compared to the prime leaves. So to investigate that guard cell SAW response, several technical challenges had to be overcome. And the first was just how to collect the guard cell enriched tissue from those prime leaves. So what I did was I carefully removed those mid veins of the leaves with a scalpel. I placed the leaves in ice water in a high-speed blender, and the uniquely strong cell walls of those guard cells allow them to remain largely intact while all the other cell types were ruptured during this blending process. That I filtered them through a 200 micron mesh, and I repeated that blending extraction process 3 times, and I could collect then this enriched guard cell tissue and snap freeze it in liquid nitrogen and store it in the -80 until I could use it. And then just to ensure that these guard cell samples that I use for all my multi-omic experiments were highly enriched, I conducted a purity assay with the guard cell samples using real-time quantitative PCR with primers for guard cell specific transcripts. And these relative expression levels of these genes were very high. And I also, the purity was further confirmed by the low chlorophyll levels of the guard cell samples when I compared them to the leaves. And in addition, the guard cells appeared to be viable based on neutral red and fluorescein diacetyl staining. So then for my experiments, I injected 5-week-old Arabidopsis plants using a needleless syringe with either PST or the mock solution without the PST as a control. And then I removed the systemic leaves for guard cell preparation with that high-speed blender. Because the guard cell materials were limited and the molecular components of guard cell SAR were unknown, I had to develop a method to profile as many types of components as possible. So I collaborated with another student in the lab to develop a 3-in-1 extraction method for identification of proteins, metabolites, and lipids from the control and primed Arabidopsis leaves. So we adapted this biphasic fractionation extraction method that was used for lipids and aqueous metabolites. And we just retained this middle phase for protein precipitation so that we could get proteins, metabolites, and lipids all from one sample. And the advantages for this multi-omic method included obviously sample conservation, but also high reproducibility and a tighter correlation between different types of biomolecules. And then I used liquid chromatography with tandem mass spectrometry for multi-omics analysis. And I just want to say that it was during this experience in my PhD that mass spectrometry became a passion of mine. And like the previous speaker said to you, I can say the same thing. When I was first able to use a mass spectrometer for my samples, I remember just feeling like Tony Stark had allowed me into his garage to use his technology. And I still have this very deep appreciation for mass spectrometers as an instrument. And I feel that they're just incredible and fascinating. And I really enjoy learning more and more about them every day during my career. And well, because of the time constraints of this talk, I can't show you all the results from my PhD, of course, but I'm just going to show you a few of the results from the metabolites of the guard cell samples of the primed and controlled leaves. So I found that the metabolites that were differentially abundant were involved in biotic stress response, in lipid peroxidation, and palmitic acid esters that were increased in abundance in the prime guard cells. So these chromatograms here on the left and the MS1 here in the center and the MS2 spectra here on the right are for salicylic acid, benzaldehyde, 2,4-DNPH, and 9-FASA are shown right here. And the chromaticons, you can see that these metabolites in the primed guard cells, that's the orange lines. When we compare them to the mock guard cells, which are the blue lines, we can see that they increase in primed guard cells. The MS1 spectra are for the precursor ions, and the MS2 spectra for the fragment ions allowed me to do the quantification and the identification of these metabolites. So then these differentially abundant proteins, metabolites, and lipids with known functions were categorized by function and number that increased, that are shown in red, or decreased, that are shown in green here, in primed guard cells. And I used a twofold change and a p-value less than 0.05 from a student's t-test as my parameters for this. And then all the identified proteins, which are shown in gray, metabolites in blue, and lipids in yellow, were identified by that multi-omic approach to be differentially abundant in the primed versus the control guard cell samples, were then categorized based on their function. And as you can see, the proteomic, metabolomic, lipidomic analyses revealed roles for ROS, fatty acid signaling in guard cells in response to these systemic immune signals. So this is really an advantage of using this multi-omic approach, because you can combine these complementary features and get a more comprehensive understanding of what's going on inside the cells. So that's what I did here. The multi-omic approach allowed me to propose roles for ROS, lipid signaling, and phytohormones in guard cell SAR. So I just want to point out some areas where my research contributed to an understanding of SAR response in guard cells. So I'm going to start over here because I found that the increase in palmitic acid in the primed guard cells. So palmitic acid is an activator of toll-like receptors, TLR2 and 4 in human cells. And it may also activate pattern recognition receptor FLS2 because FLS2 has a functional homology to TLR2 and 4. And remember that FLS2 binds to that molecular signal, the MAMP flag 22 from the PST. So additionally, salicylic acid was increased in the prime guard cells, and SA binds to its receptor, NPR1, non-expressor of pathogenesis related 1, and it's a key regulator of SAR. So ultimately, SA acts on these membrane anion channels, driving the water out of the cell and closing the stomata. There's also intercellular accumulation of ROS that can lead to oxidative stress, but oxidation of fatty acids and lipids can help reduce that oxidative stress. And I identified several compounds that had a differentially abundant compounds that had antioxidant properties in the primed guard cells. I also identified these phosphatidylethanolammines, or PEs, in the primed guard cells that were differentially abundant. And PEs have a conical structure that's specialized, and it can cause membrane alterations in those primed guard cells. So just briefly, I want to talk a little bit about the research I did as a postdoc at NC State, which involved using mass spectrometry to identify a specific type of mobile signals in plants called peptide hormones. Sometimes in the literature, they're also called small signaling peptides. And if you remember, the mobile signals for ISR-induced systemic response are unknown, but they may include these small peptide hormones. So for my postdoc, I began working with this model legume called Lotus japonicus, and I developed a method for identification of small signaling peptides. I collected the foliar and root tissues and also vascular fluids from these lotus plants, and I modified a published method for peptide extraction and used undigested samples that were filtered using this pre-washed 10 kilodalton filter to collect the small endogenous peptides in the flow through. And this portion also has a lot of small metabolites, which makes mass spec detection very complicated. And then the proteins that were bound to the 10 kilodalton filter, I digested with trypsin and I collected for proteomic analysis. So both endogenous peptide portions, the endogenous peptide portion and this tryptic digested protein portion were analyzed using liquid chromatography coupled with tandem mass spectrometry. And the data was analyzed using PEAKS software. So PEAKS allows two types of peptide identification. The first is matching the peptide sequences to the Lotus proteome database, but the second is identification of de novo peptide sequences from the MS2 fragment ion spectra and detecting that are detected by the mass spectrometer. So just to conclude with some of my past and looking to the future. So in conclusion, my PhD research, I was able to add information to key knowledge gaps in the field of systemic acquired resistance and stomatal immunity. So some of my key findings were that primed stomata exhibit this altered response to bacteria, that the stomata priming decreases the bacterial colonization of the leaves, that there's roles for ROS and fatty acid signaling, and that palmitic acid may play a role as an activator of FLS2. I wasn't able to go into detail about it here, but there were two long-chain fatty acids, including stearic acid and behenic acid, that I identified as putative lipid signals that could be transported by that lipid transfer protein DIR1, defective in induced resistance 1. And the DIR1 chaperone may activate SAR responsive biosynthesis of amino acids, and it also relates to particular wax esters in the leaves. By using this multi-omic approach, it's an untargeted approach, I was able to identify some candidate molecules for a targeted approach analysis by reverse genetics. I also, during my PhD work, I did a lot of method development. So I developed this 3-in-1 extraction method from the guard cells. And during my postdoc, I developed this method for protein and peptide extraction from plant tissues and also from vascular fluids. I really want to thank everyone for giving me this opportunity. I just want to say, I have a lot of experience now with mass spectrometry, both in academia, and now I'm moving to industry, starting my new job. And I also have this passion for teaching, mentoring, and also doing research. So I also have a strong background in promoting diversity. And my goal is I hope to find a tenure track position that will allow me to teach undergraduate and graduate students, but also pursue my research interests. And with that, I want to thank you, and I want to offer you time for any questions or just to give me any feedback from my future goals. And thank you so much for giving me this time.
Thank you, Lisa. Great. I really appreciate that you took the time to explain things for people like us who don't work on plants. And even though I really love the plant world in general.
Important. I thought it was important.
Yeah, it is.
I cut out so much of my research, but if I didn't do that, I don't know how people.
I mean, FeMS is all MS field, right? So it's really nice that you thought of this. And does anyone have a question for Lisa?
I have a question. Hi, Lisa, congrats on your work. My question is if to analyze proteins, if you needed to put any proteolysis inhibitor in your samples, or and if you did, if it was before or after destruction?
Yeah, we did add an inhibitor. So you're talking about that 3-part extraction, right? And then we precipitate from this middle layer. It's not really a layer, it's like a scum layer. We remove the top layer and the bottom layer, and then we leave the middle layer, and we put in like an inhibitor. We precipitate out from there, and that's like a regular protein prep, right? From that fraction.
Thank you. It was just that question because I was like, at what time you put this, because people normally use, and I was wondering if I need to put before start extracting.
The whole protocol is actually published, so you can just, you could just go to my LinkedIn page, or I think on my Twitter, maybe not my Twitter page, but definitely my LinkedIn page, or you can just Google search me too and find me, and we have that whole protocol step by step with like a diagram from where you add each thing. It's a protocol paper.
Thank you very much.
It's very kind to share your standard, your SOPs. I'm in the same position for a total different thing.
Published 2020, I think it's from almost 3 years.
Yeah, you never find as much details in publication as you would in SOP. So I really appreciate when people share their SOP. That is really detailed as to what to do. We had 2 other questions. Let's go very quick. I don't want to go past the next 9 minutes. And so I will, Behnaz, you want to ask something?
Yes. Thank you so much for your talk. It was great. So one question I have is for your slide 17. You were talking about the antioxidants that you have. You figured out that there are some valuable antioxidants in your fractionation. So my question is that, have you been able to purify them and see what if it is a possibility to use it in pharmaceutical science, for example, drug delivery system, or healing, for example, or any wounds, for example? That's my question.
If I did a second PhD, that's what I would do. So now that I took this industry job, this is more targeted approach, right? Like you're talking about, like biomarkers. So during my PhD work, it was all like untargeted, like discovery based. And I think those are like the preliminary steps before you can get to this targeted research, which is so important because you're validating those things that you discover that are differentially abundant, but you have to find out what they're actually doing. And these are like antioxidants that are differential. So I assume that they're like quelching this ROS burst, right? Because with this ROS burst, you could have cell death, right? Apoptosis, but it has to be quenched by this lipid peroxidation and antioxidants that are produced in the cell. But we have to validate this. So I think that's the next graduate student. I don't know. So it wasn't me. I did my 5 years. So I don't know how to say that, but yeah, there's always more research that can be done. And it's a fascinating field of plant immune responses and just plant cell biology.
Thank you. Vinny, one last question and then we have to
Yes.
close.
Hi, Lisa.
Hi.
Thanks for the wonderful talk. So I was actually curious on your that intrinsic peptides story, that you have identified a number of intrinsic peptides in the ISR system.
Okay.
So I was curious to know whether did you identify any of these cyclic peptides?
Cyclic peptides, like those
Yes.
peptides that are made from like some little non-coding sequence. So I only spent 1 year at NC State. And so I started to classify those different peptides that I found. So it was mostly just method development of how we can honestly just pick those small peptides out of, like, okay, so maybe if you haven't worked with plants, plants have maybe 1 million small metabolites as well. And if you're taking this flow through fraction, you're getting all these small metabolites and all these small peptides. And these small peptides, many of them are also singly charged. So, you know, if you do proteomics, you would just exclude those singly charged precursors, exclude them from fractionation, right? Or I'm sorry, not from fragmentation, right? So those wouldn't be in your MS2 spectra. But you know, in this situation, you have these things are usually, they're less than 20 amino acids long. Some are less than 10 amino acids long, and they're often singly charged, and they're mixed in there in your sample with these small metabolites, which are also singly charged. And it's just a method of picking those out from your spectra and identifying those, and not, you know, if you're doing a top 10 pick or a top 15 pick, you're going to pick whatever's the most abundant. And a lot of times that will be small metabolites, not the peptides, because peptides are often, especially if they're signaling peptides, a signal is usually something that's low abundant. So a lot of what I did was method development for how we can get to that point of finding them in the sample. And then, you know, there will be identification downstream of that, right?
Thank you.
I hope I answered that okay.
Yeah.
Okay.
Thank you. I don't see any other hand raised. So I think I will close this session. Thank you very much, everyone, for being here, and for our speakers to show what they have been doing. And I really wish you all the best of luck for the future. And please do not hesitate to have a look at this underrepresented scientist directory. You can actually register yourself there, right? And maybe it's good because you can also find people in your field and in fields you're interested in to reach out. So with that, Amber, do you have a final word or?
No, great. Congratulations on your first event.
Yeah, oh yes, yeah. And
FeMS.
actually I realized I forgot to introduce myself. So that will be my last word, because it's not under my name. I'm logged in, so no one knows me here. I'm Julie Courreau. That's the very French pronunciation of my name. Julie, working in Athens, in the University of Athens, in metabolomics, actually, clinical metabolomics, and now proteomics as well. So yes, and next time I promise I will introduce myself first and not at the end of this recording. I will stop the recording now, and I wish everyone a good evening.
And before you shut the meeting, Janet, I missed you when you introduced yourself. I wonder if you could introduce yourself again.